All right, welcome everyone. Today's session of the American and Floral Endowment, or AFE, webinar series for thrips and botrytis research. Today is the second of a three-part research series of the thrip series. I'm your webinar host, Dr. Garrett Owen, Assistant Professor of Greenhouse and Controlled Environments at the University of Kentucky. Today, we'll be hearing the second part from Dr. Rose Buitenhuis, Research Scientist of Biological Control at the Vineland Research and Innovation Center in Lincoln, Ontario, Canada. Before we get started, I have a few housekeeping rules I'm just going to go over. Is that when you all came in, you were muted, and that we ask that you submit any questions through the Q&A chat at the bottom right of your screen. We will answer any questions at the end of today's presentation. I do want to go ahead and say that these sessions are being recorded and will be edited for public posting in the spring of 2021, and that these presentations materials are copyrighted and should not be distributed or published without AFE written permission. So before we start hearing about some of Rose's research, I'm going to go ahead and talk about the Thrips and Botrytis Fund. In 2017, a special research campaign was established to address and control management of botrytis and thrips. Losses from these pests affect all segments of the industry every day. Botrytis and thrips are challenging to control using currently available pesticides and management strategies. In 2018, AFE reached a goal of $1.5 million pledged to the industry support to conduct new and innovative research leading to effective control procedures and management strategies. With these funds, AFE has been able to support 8 multi-year research projects. The research will lead to reduced losses and higher quality of flowers and plants. So at this time, the American Floral Endowment and all researchers would like to thank all of the organizations that have made contributions in support of these important research priorities. So without further ado, I'm going to go ahead and turn it over to Rose so we can hear about the opportunities and challenges of biocontrols of thrips. Rose, the floor is yours.
Thank you very much. And thanks again. Hopefully we'll have a few people who were also at the last webinar. Welcome back. Last webinar, I talked a lot about like why are thrips such a pest and why are they so hard to control. Today, I would like to talk more in a more positive way, I guess, about the opportunity for use of biological controls. So I often hear the question, is it possible to manage thrips using biological control as the main component of your IPM strategy? And from where I am in Canada and my research and my knowledge, I would say yes. Growers in Canada have really fine-tuned biological control against thrips, and they're successfully dealing with pesticide-resistant thrips. The next question is, can any of these strategies be used in other parts of the world? And especially, I get the question through AFE, can it be used in Colombia? This presentation, I will try to discuss the similarities and the differences between the floriculture greenhouse sectors in these two countries. I will try to also throw in a bit of knowledge for the US, because you'll see there's a lot of similarities between Canada and the US. And I'll talk about the tools that are available and how to take the first steps towards a biocontrol-based IPM strategy against thrips. So here's a visual summary of thrips IPM programs in Canadian floriculture greenhouses. And this example is for potted chrysanthemum, but the program is easily adjusted to other crops, vegetable or floriculture crops, or even sometimes new crops like strawberries and stuff. It shows the tactics that were used from sticking of unwooded cuttings, kind of here on the left, to all the way to production, to the sale of the final product. At the unwooded cutting stage, I will talk about dipping, but also there is a release of predatory mites in the Canadian system. When the plants come out of the misting house, they get a whole load of biocontrol agents, including some soil predators. Here we have Stratiolelaps and Ethita, also known as Hypoaspis and Dolosia, and nematodes, spranches, releases of or applications of biopesticides as Bovaria, and again, broadcast of predatory mites several times or weekly. When the plants get spaced, put further apart, they receive sachets of predatory mites, maybe some more applications of biopesticides, and other predators like orioles come into play. And then finally, at the point where it's ready for sale, if there's still any residual thrips populations, you can do a cleanup spray with a pesticide. And because you haven't used pesticides throughout the production cycle, the likelihood that it will work and that the thrips are not resistant is very high. Also, throughout the production cycle, you'll see strategies like mass trapping with sticky cards or sticky tape is taking place. So that's the program as it seems to work well for Canadian growers. Now, if you're in the US, you will have a lot of these biocontrol agents to your disposal. Also have a lot more chemicals to your disposal. So I think one way or another, you will be able to make this work. Now, not all these products are available in Colombia. There are a few biocontrol agents available. Phytosalus persimilis and Neocelus californicus are both predators of spider mites, and they will not really contribute in any significant way to thrips control. On the other hand, Bovaria bassiana is an entomopathogenic fungus that can play a very important role in thrips control. And everywhere where you saw that like green spray icon in the infographic, that's where Canadian growers use Bovaria bassiana. I think in Colombia, there's also mineral oil available. I mean, technically, it's not a biopesticide. It's more like a reduced risk pesticide, but it's a very useful product to integrate with biocontrol. And I will talk more about it in the section on dipping. So if you start from unrooted cuttings, you will have the best success with your IPM program if you can start clean. And this counts for any crop. If you start clean or free of pests, your IPM strategies will work better because you don't have to play catch up. For crops started from cuttings, this can be achieved easily by dipping unrooted cuttings. And this was research sponsored by the AFE. I'll talk a bit more about it. So for cutting dips, you take the cuttings out of the bag, as you can see here in the pictures. You spread them loosely in a perforated tray. Don't pack them too tightly because you want the cuttings to get covered completely. So for this, they have to be able to float a bit. You can cover this tray with a second, that's not what I meant to do, with a second perforated tray, and then completely immerse the thing in the dip solution. For small quantities of cuttings or very tiny cuttings, some people prefer to use a colander or a sieve, and it will work fine as well. The goal really is to get complete coverage of the cuttings. So in this research, the products we investigated as dips were biopesticides or reduced risk products such as oils and soap. And the reason why we chose these products is that they have less issues with pesticide resistance. They also leave minimal residues, and they're highly compatible with biocontrol agents. So after dipping, the cuttings are stuck in a substrate as usual and grown again according to normal production practices. So in this graph, you see the main results of the research. I mean, we did many trials in the lab and also in commercial greenhouses, but here I'll only show you the result of a research greenhouse trial. So we tested different cutting dips, the effect of different cutting dips, Bovaria bassiana, or a mineral oil product, and it's called landscape oil here. And those are the solid lines in the graph. We also combined dips with the release of predatory mites, and these treatments are the dotted lines. The graph shows the number of thrips per pot over time, right here on the x-axis, over the course of 4 weeks. And as you can see, the blue lines are the control treatments, and the populations went up really quickly, really fast. Even with biocontrol, so the predatory mites, populations of thrips still went up in week 4, pretty high. In contrast, both dips, the yellow and the red lines, decrease the number of thrips significantly. The yellow lines in the graph are on top of the red lines, actually, so that's why the red line is a little bit harder to see. And also the dotted lines, if you follow up the dip with biocontrol, the number of thrips was the lowest. So the take-home message of this trial is that cutting dips basically set the clock back on thrips populations and give time to biocontrol agents to start working. Now, an added bonus in this trial was that we saw that landscape oil also reduced spider mites. The cuttings were accidentally came in also with spider mites, and we definitely saw that landscape oil both reduced thrips and spider mites. So that was an extra bonus. Now on to entomopathogenic fungi. So Beauveria bassiana, we use it in the dip, but it's a very good product for thrips control, both preventative or even curative. And you can replace several pesticide sprays with Beauveria sprays, actually. You have to get the fungal spores in contact with the thrips, so good spray coverage is very important. That means apply it in low volume, so that's basically a fine mist that reaches both the upper side and the underside of the foliage, and you want to avoid runoff, because when it drips on the soil, it doesn't do much for thrips. Recent research at Vineland showed also that cold fogging of Beauveria is possible. And in cold fogging, the spores are not exposed to heat when the liquid is vaporized, as opposed to pulse fogging. So in cold fogging, the spores are not heated up, and the coverage of the foliage was excellent and gave very good control of thrips. Finally, there's some formulations of entomopathogenic fungi like Beauveria bassiana or Metarhizium anisopliae, MET52, as a granule formulation. And you can apply this to the substrate, where it will help control thrips that pupate in the substrate. So for a country like Colombia, that's all the commercially available products. So what about the rest of the stuff I showed in the infographic? If you can't buy it, where would you get some of these? Well, there's many predators that actually naturally occur in the landscape and that are very similar or even the same to the biocontrol agents that are available in the US and Canada. In Colombia, there's Aureus insidiosus, which eats thrips, aphids, and some Lepidoptera as well. There are native predatory mites that live on the foliage or in the substrate. And I'm not sure if rove beetles like Dolosia or Athida are in there, but I definitely know there's generalist predators like lacewings, Chrysopa, syrphid flies, and lady beetles. So if they occur naturally outside, the question is, how do you get them into the greenhouse to control thrips? Here you see a picture of me, and I'm looking for Aureus in the vegetation around one of the greenhouses that I visited in Bogota, in Colombia. So at the time, this greenhouse had a strategy to control all the weeds around the greenhouse to reduce the habitat for thrips, so that they would have less thrips pressure from coming in from outside, and it makes a lot of sense. However, the flowers that I inspected still had a lot of thrips, and you can see them here on my hand, all these little dots where unfortunately were thrips. And I could not find any predators. Like if I would do this in Canada, I would often find things like Aureus. And I suspect that the chemicals that were applied around the greenhouse, the herbicides, did kill or repel the naturally occurring biological control agents. So that was an example where they could probably not count on predators coming in from outside. If you are able to find native predators and collect them, there are relatively simple ways to mass rear them on the farm. And you can do this also with some of the biocontrol agents that are commercially available if you want to rear them yourself. And here are two examples from the literature. These are the articles. If you input them in Google Scholar, you will be able to find the original article. So Aureus can actually be reared in jars like this, using flower moth eggs or Ephestia eggs as food, which is, again, you can buy this from some of the supply companies, and also pieces of plants as an oviposition substrate. So that's all in this jar here. For predatory mites, you can rear them in these plastic boxes, and their prey are these storage mites. Both systems are relatively simple once you have the right organisms, and they will yield high numbers of predators. There's also other rearing systems for other biocontrol agents. So if you want to know more about this, let me know and I can point you in the right direction. Lastly, if you can't rear them, you can attract and maintain populations of biocontrol agents on habitat plants. And you may have heard about habitat plants from another researcher that's supported by the AFE, Dr. Margaret Skinner from the University of Vermont, and she has done a lot of work on this. The Twitter post I'm displaying here is from Ronald Valentin. You can see all the banker plants here, all these different plants here are all banker plants that are grown at a large American greenhouse, Metrolina. And these plants are colonized by different biocontrol agents. So either they're inoculated with the commercially available ones or they come in from outside. There's mullen plants that are colonized by Dicyphus, a predatory bug, a mirrored predatory bug. There's ornamental peppers, the purple plants that are good banker plants for Aureus insidiosus. And the white plants are alyssum plants, and Aureus and syrphid flies love these alyssum plants. So these plants basically become breeding factories of biocontrol agents. And when they're throughout your crop or when they're inside your greenhouse, the biocontrol agents will move into the crop to manage your pests there. So there's a lot of interest now also in doing research on what plants to grow in the landscape surrounding the greenhouse so that you attract and encourage the establishment of naturally occurring predators. And that will be very interesting research to follow and see if it will contribute anything to biological control inside the greenhouse. Now, as you will hear in JC's presentation next week, there is still a big role for chemical control of thrips. Even in Canada, you saw that cleanup spray, or if the biocontrol program gets off the rails, chemical intervention is still necessary. However, if you mainly rely on biological control, this will allow you to reduce the number of applications and have higher chances of the thrips not being resistant. However, balancing or using biocontrol and chemical control together, integrating the two, is not easy. Even if you stop spraying today, there are still many ways that pesticides can interfere with your biocontrol program. First, there's many pesticides that leave residues on the crop and on the greenhouse structure, and those can persist for a very long time and negatively affect your bios. Also, if you've been using broad spectrum pesticides, they may have been controlling pests you didn't even know you had, or that previously did not cause any problems. Now, when you stop these sprays, these secondary pests may cause an outbreak. So that's something that can occur. But integration of biocontrol and pesticides is possible. So you have to consider well which pesticide you want to use. And then it can reduce pest populations before releases of biocontrol agents, so that they have a better chance of success. Or they can clean up the plants before they're sold, or they can be used as spot sprays to treat hotspots of pests without affecting the biocontrol program in the entire greenhouse. Now, a big help for choosing the right pesticide are these side effect databases that you can find on the websites of several biocontrol companies. Here I took the covered side effects database, which is one of them, and I put in some of the pesticides that are registered in Colombia, because I did this talk also at the CIFLOR conference. So that's why I took these as an example. But here are some of the pesticides that are available. And vertically, you can see some of the main biocontrol agents that are available in Colombia. So the output is basically a table. And you can see the cells are filled with a number, a color-coded number, which is the effect of the pesticide on the biocontrol agents. And it goes from compatible to toxic, green to red. And also you see these numbers here are the persistence of this effect in weeks or days sometimes. A common misconception is that you only have to check the compatibility of insecticides and acaricides. But some fungicides and other chemicals can also have devastating effects on biocontrol agents and disrupt your carefully prepared biocontrol program. So it is very important to check. Now we'll go through a few examples. So here's an example in this column here. You can see this pesticide causes less than 25% mortality in the selected biocontrol agents. And this is indicated by a green number 1. This product also has 0 residual effect. So that's very advantageous. You have to note, however, for some biocontrol agents like Aureus, there's no data right here. So we don't know. You can't assume that if it's good for one, that it's okay for the other as well. And the other thing is also, this table only gives you information on the direct mortality of the biocontrol agents, how many die. It is still possible that this pesticide affects the searching and predation capacity of biocontrol agents. And we call that sublethal effects. So even if they do not die, the biocontrol agents, when they are used or when they come in contact with this pesticide, may not give you the level of biocontrol that you are expecting. But overall, green means compatible. Now, this example shows 2 pesticides that cause more than 75% mortality in the selected biocontrol agents. You can see here it's a red number 4, and it means more than 75% mortality. These products will wipe out your biocontrol program. However, the first one here has a residual effect of only 1 week. And the residual of the second product stay toxic for 8 to 12 weeks. So if you have to choose, if possible, you should choose the product with the shortest residual so that you can rebuild your biocontrol program right away as soon as possible. Now, these pesticides are considered not compatible. However, you can still use them, like I said, if the residual time is short, or if you use them as a spot spray, just in certain areas, so that you don't spray the whole greenhouse and not wipe out your entire biocontrol program. Or you can use them as an end of production cleanup spray, because the plants will be sold and the biocontrol program is done. Now, the final example is of a pesticide that is compatible or not, depending on how you apply it, on the application method. So here you see a lot of red and orange, which means toxic. And here you see quite a bit of green, which is nice. So that means compatible. And it really is the sprays are toxic. Right here, this is the spray, SP means spray, but the drenches are compatible with certain of the selected biocontrol agents. Not all, because here you see Aureus insidiosus is still killed by this. And that's probably because Aureus sometimes punctures the plant a little bit to get some water from the plants. So that's how it may ingest some of this systemic insecticide. So that's it for integration of biocontrol and pesticides. And we've talked about control agents, biocontrol for all this presentation. But integrated pest management is way more than that. And if you don't pay attention to the effect of the plants and the effect of the environment that you grow them in, you're setting up your biocontrol program for failure. And we talked a little bit about this in my last presentation. It's really the systems approach to thrips IPM that will make it all work. In the last few slides of this presentation, I will explain a few ways you can make your production system more resilient against pests like thrips. The first of these strategies is mass trapping. You saw it featured very prominently in our infographic on one of the first slides. Mass trapping is one way that you can remove a lot of thrips from the greenhouse. And these two graphs are research done by Dr. Sarah Jendrisic. She's with the Ontario Ministry of Agriculture, Food and Rural Affairs. And she found that most yellow sticky cards or rolls catch more thrips than blue ones. I mean, you normally hear that blue is more specific for thrips, which is still true, but certain yellow cards, especially the first bar you can see, capture way more thrips than the blue ones. And this depends on the company making the cards or the rolls, as the yellows and blues are slightly different hues. So like I said, blue traps are considered more selective for thrips, but the advantage of yellow is really that you can monitor thrips and other pests at the same time and see if there are any biocontrol agents flying around. Only if you start trapping a real high number of biocontrol agents, I would say consider changing from yellow to blue, because then you start removing too many biocontrol agents from your greenhouse. But a few here and there will just tell you that the biocontrol agents are there, which is nice confirmation. So the traps should be placed just above the crop canopy. And inside vented greenhouses, so where the ventilation comes from the side, you also should place a lot of traps close to the vents to trap the pests that are coming in just before they land on the crop. We have ongoing research that's looking at the option to combine traps with thrips lures, such as the aggregation pheromone or methyl isonicotinate. And some research shows that significantly more thrips are trapped on traps with lures, and other studies show only a slight increase. So there are other factors at work that we don't know exactly what they are. If you want to try lures, it's probably best to do an experiment for yourself to see if lures will work in your greenhouse. And of course, if they work or if they don't work, I would be very interested to know about it. Another of these arrows in my systems approach graph talks about the plant. So consider what plants are you growing. Greenhouse crops have traditionally been bred for higher yields in vegetables or a nice appearance in ornamentals, with little regard for susceptibility to pests and diseases. Now, the breeding industry is increasingly investing in the development of pest resistance. So the results are promising, even in ornamentals, where thousands of species and cultivars are grown. For example, this graph is research from 2014, And these are all different chrysanthemum varieties. On the y-axis is how much thrips damage they will show. So some of these varieties showed no thrips damage, while the same exposure to thrips in other varieties showed really high thrips damage. So you would probably want to choose some of these plants and maybe pass on some of these. And it was very promising in this research as well that there was cross resistance. So increased resistance to one pest resulted in enhanced resistance to another pest, like leaf miners or spider mites in this case. So that is very promising, especially because it is resistance against several pests. I think hopefully we'll see more of this kind of research for other crops as well, especially ornamentals. And now that we have better molecular technique, like breeding techniques such as marker-assisted breeding, this will make selection of specific resistant traits easier, faster, and cheaper. Now, this breeding is one thing, that's like the genetic resistance, but are there things that you can do to your plants that will make the plant less attractive to pests? And there are studies that suggest that feeding plants with a lot of nitrogen makes them a better food source for pests like thrips. And traditionally, a lot of crops are over-fertilized, so there is actually room for reduction of fertilizer without effects on plant quality. So sponsored by AFE, again, we are currently doing trials to see if we can use this as a component for an IPM program. And our hypothesis is that reducing fertilizer will slow down thrips population development. And this is because nitrogen is transformed into proteins and amino acids, and that's what thrips need, just like we need nutritious food. Thrips as well thrive on plants with high nitrogen. So in our first trial, we saw that lowering inputs from 200 ppm of nitrogen to 50 ppm of nitrogen resulted in quite almost half the populations of thrips, especially in, you can see here, sorry, I should explain this graph. This is the number of thrips per pot in week 5, week 7, week 11, and week 14. And the different colors are the different fertilizer regimes, from 25 ppm of nitrogen to 300 ppm of nitrogen. So here in week 11 and week 7, you can see that the lower nitrogen feeds resulted in less thrips. Now, this research is still in progress. We repeated the trial several times, and the results were all the time slightly different. So we're not at the point yet that we can really formulate recommendations, but definitely there is a difference between low and high feed. Now, we saw, and you can see in this picture here, that plant quality started to suffer below 100 ppm of nitrogen. So another part of the study was to see if there was a role for things like biostimulants to improve plant quality at low fertilizer levels, that the biostimulants could help the plant cope with low nitrogen levels. So that's still all ongoing. I just wanted to show you some preliminary results here. So wrapping it all up, implementing a biocontrol program against thrips is not easy. If you're in Canada and the US with all the tools available, or in another country where you may not have all these tools to your disposal, even in countries where all the biocontrol agents are commercially available, developing a reliable and cost-effective biocontrol program takes several years. So it is a process that takes a lot of patience, faith, and perseverance, And things will go wrong several times before they start going right. So don't give up too easy. You are on the right way. One of the biggest risks to biocontrol programs comes from pesticides. And choosing the wrong pesticide to address a problem can completely wipe out your biocontrol program. So that's why I spent so many slides on explaining the side effects table so that you can choose the right pesticide. However, when you succeed in biocontrol, setting up a biocontrol program that works for you, you will reap all the advantages, which include sustainability, worker health, safety, better crop quality, and you will have a resilient system that can deal with resistant pests, which is basically the driver behind implementation of biocontrol, especially against thrips. So with that, I come to the end of the biocontrol portion and the systems approach portion of this presentation. You can see here my email address. If you have any questions, don't hesitate to send me an email or look me up on the Vineland website. I'll give it over to Garrett again to moderate the questions. But thank you for listening.
Thanks, Rose. That's very interesting. So if anyone has any questions, you can go ahead and drop those questions into the chat or into the Q&A, and we will entertain those as they come in. So Rose, I have a question based off the research that y'all were looking at in terms of reducing the nitrogen input and thrips populations. So by doing that, reducing the nitrogen application or delivery to the plants, there is what I would say, reduced quality in the overall plant architecture. So if one wanted to reduce the inputs, but say increase greenhouse temperature to maybe overcome some of the negative plant growth effects from withholding that nitrogen, would we see a difference in the population as well because we've increased, say, the greenhouse air temperature?
Yeah, if higher temperatures means faster thrips cycling, but if you use biocontrol, biocontrol will be sped up as well, so that may make up for it. Now, the reason why I was looking at reducing nitrogen is that you actually have room to reduce the nitrogen quite a bit before plant quality starts to suffer. So I really wanted to see how far do you have to reduce it until you see an effect on thrips. And we're playing around at 100 ppm of nitrogen level, which in our case still yielded nice plants, a good architecture, a good number of flowers as well. So if we can play around that level, I think it may be a way to slow down the thrips populations without having to do other things like increase the greenhouse temperature.
Okay. And I know that there's a lot of alternative greenhouse substrates being used. And some of the research I did with wood fiber a couple years ago, trying to look at nitrogen immobilization and nitrogen drawdown. So there was the initial thought that you have to provide a little bit more nitrogen to overcome that effect of the nitrogen to carbon balance. So if one was using, say, a wood fiber based substrate or wood component substrate, say, and having to apply instead of 100, but 125 or 150, would you say, based off some of your preliminary work, that would still influence the population?
It all depends on what the organic nitrogen levels in the plant are. So that's why I don't want to focus too much on what you feed. You really have to see like how much nitrogen do you end up having in the plant. And we did plant sap analysis as well.
Okay.
And we we will be able, hopefully, to give an indication what those nitrogen levels were.
Okay.
Now, nitrogen is not the only nutrient that can have an effect on pests, the other nutrients as well. So this is just the start of a whole new research area, I would say. I mean, you hear a lot of interesting things about silica and other calcium. So there may be other nutrients that are very important as well.
Okay. Yeah, because I was wondering if there was any correlation maybe between plant sap that someone could measure in-house, such as nitrate, or having leaf tissue analyzed and correlate that with maybe thrips population and cultural inputs.
Yeah, definitely.
Interesting. Awesome.
We chose the SAP analysis because the thrips are feeding on the cell contents. While when you do tissue analysis, it's more, you get more of the solids in there. I hope I'm not a plant physiologist. I hope I got this right.
Yeah.
But anyway, that was our thinking.
Okay.
So, yeah.
Excellent. So we did have a question come through the chat, and this is from Juan. And basically, the question is, if thrips are way above the economic threshold and pesticides needs to apply to help thrips populations go down, what are the best options, and for how long do we need to wait to go back on biologicals controls?
Yeah, so this is the perfect situation where you would go to this side effects table of one of the companies. I showed the example of Coppert, but BioBest and a few other companies have similar tables. So you would put in the options that you have of the pesticides that you're considering and the biocontrol agents that you want to keep around or that you want to release right after you apply. And then just look which ones has the least toxicity to your biocontrol agents and the least persistence. So I can't off the top of my head kind of recommend like, oh, you have to use this product, but yeah, just use the side effect table. That's exactly what it's for.
Okay. So I don't see any other questions coming through the chat or Q&A at this time. So with that, I think we'll go ahead and start wrapping up. So again, let's thank the researchers, Rose, for participating today, and all the individuals participating and listening, and supporters for all of you tuning in today. And that if you have any additional questions or would like more information, please contact AFE's research coordinator, Dr. Terrell Nell, at tnell@afeendowment.org. And all of this information was just dropped in the chat. So if you want to extract his email and ask him to share some of this information, you can do so in your free time. And that for future research updates and any other floral industry resources, you can visit endowment.org. Once again, these presentations and materials are copyrighted and should not be distributed or published without AFE written permission. The videos will be released in spring of 2021, but not to the public until then. So for you all that attended, you'll get the first viewing. And sponsors and supporters will have access to these video recordings at the end of the series. Thank you.